Welcome back to a new video of Sunanda's tutorial. In this video we will discuss about the cyanobacteria. Cyanobacteria is present in your UG Major 1 C1 paper as well as in minor botany syllabus. So without wasting time, let's start. So at first, let me tell you some general characteristic features of blue-green algae or cyanobacteria. Actually, the cells of cyanobacteria are prokaryotic in nature. It is the first point. Second one, they completely lack flagella. Okay, they lack flagella. Even their reproductive cells are non-flagellated. Okay, so the locomotion is absent, but they show some gliding movement. Okay, next the pigments, they have some characteristic or unique pigments like C phycocyanin, C phycoerythrin, chlorophyll A, beta carotene, and xanthophyll, a special kind of xanthophyll called mixoxanthin and mixoxanthophyll. Okay, then there are no membrane bound chromatophores. Actually totally they lack plastid. They have pigments, green pigments, but they lack any plastid. The unique food storage compounds, they have unique food storage compound called mixophycean starch and a protein called cyanophycin. Okay, remember these two terms, mixophycean starch storage product and one protein called cyanophycin. Then they possess a special cell which help in nitrogen fixation called heterocyst. Blue-green algae or cyanobacteria possess a specialized cell called heterocyst. Heterocyst help in the nitrogen fixation. Sexual reproduction is completely absent, but in some species genetic recombination is observed. Okay, next these blue-green algae or cyanobacteria are widely distributed in aquatic environment, in different types of aquatic environment, in marine environment as well as in freshwater environment, in flowing water, in stagnant water. So they are widely spread, widely distributed. Okay, a typical example, a marine blue-green algae called Trichodesmium erythraeum. Okay, remember this, the name. Trichodesmium erythraeum. Okay, so in which the red pigment, that is C phycoerythrin, predominates, and this species mainly grow in Red Sea, and this species is typically responsible for the red color of the water of Red Sea. The Red Sea appear as red color because of this species of blue-green algae or cyanobacteria. Okay, so the species of cyanobacteria can grow in very low temperature. They can grow in hot springs. They can also live as symbiont. Some blue-green algae grow in the root of cycas because cycas root, cycas coralloid root have blue-green algal zone. Then they also grow in the thallus of Anthoceros bryophyte. Then annabena occur in cavity of the fronds of Azolla. Azolla is a water fawn. So they also live as symbiont. Okay, and next, the morphology of cyanobacteria, they may be unicellular. That means they sometimes they live individually, sometimes they form colony, sometimes they are filamentous structure, they have filamentous structure. Each filament consists of a sheath of mucilage and one or more cellular strand called trichomes. So a single filament of cyanobacteria called as trichome. Sometimes more than one filament are present called as trichomes. So surrounding this trichome, mucilaginous sheath is present like this. Okay. Single trichome filaments may further be of two types. One is homocystous. This filament or trichome can be homocystous, that means undifferentiated. All cells of trichomes look alike, look same. Okay. But there are some species and the trichomes are heterocystous, that means differentiated, having heterocyst. Some cells are heterocyst and some are normal, appear in nostoc. Okay. And homocystous trichome appear in oscillatoria. Okay. Spirulina has spirally coiled filaments. Okay. Spirulina has spirally coiled filaments. Colonies develop in some cases. For example, in nostoc, colonies are developed. Okay. So these are some morphology, external features, how they live, in which habitat they live, how they appear, and their body is called trichome. There are some trichomes which are homocystous. The cells, involved cells in trichomes are undifferentiated. Some have heterocyst and their trichomes are called as heterocystous because their body is differentiated into normal cells and some specialized cells called as heterocyst. Next, flagella are completely absent but they show gliding movement. I have already told you about that. Then the name oscillatoria has been given to a common blue-green algae on the basis of pendulum-like oscillating movement of its anterior region. Their body, anterior region of the body, so pendulum-like movement. So the name given to that species is oscillatoria. Next, like bacteria, the cells of cyanobacteria also consist of a mucilaginous envelope which is present exterior to the cell called as mucilaginous sheath. Then inner to it cell wall is present. Next, plasma membrane, then cytoplasm. So at first the sheath, the whole cell of cyanobacteria is covered by hygroscopic mucilage-like mucilaginous sheath. It protects the cell from external unfavorable conditions. Okay. Next, cell wall. The cyanobacterial cell wall consists of 4 layers called as L1, L2, L3 and L4. So L1 is the innermost, okay, which is present exterior to plasma membrane. It is the plasma membrane. Then L1 is present inner to it. Next, outer to L1, L2, then L3, next L4. Okay. Next, so L2 is electron dense layer. It is made up of mucopeptide and muramic acid, glucosamine, alanine, glutamic acid and diaminopamelic acid. L2 provides the shape and mechanical strength to the cell wall. Okay. Next, L3 again electron transparent layer. It is comparatively thin than L2. L3 is comparatively thin than L2. Then the outermost one is L4. Okay, L4, which is a thin but electron dense layer. It appears wrinkled and is undulating or convoluted. Okay, all the layers are interconnected. All of them are interconnected with each other by plasmodesmata. Plasmodesmata are the connecting link between those layers. Okay, next, so it is the diagram of cell envelope of Cyanocystis species. So here you can see surface layer, then outer membrane, then peptidoglycan, periplasm, inner membrane. So in this way 4 layers appear on the cyanobacterial cell. Okay, next, the chemical constituent of cyanobacteria and gram-negative bacteria is presence of mucopolymer, which is made up of five chemicals, 3 amino acids and 2 sugar. Okay, similar ratio in this constituent is found in E. coli also. However, in some cyanobacteria such as Anacystis, Formidium, then Chloroglia, the amino acid and sugar are found in different ratios. Okay, but chemical constituent is same, but ratios are different. Moreover, diamino acid is common in all prokaryotes. In addition, lipid and lipopolysaccharides have also been detected in some cells of cyanobacteria. Okay, next. Next is the plasma membrane. The cell wall is followed by a bilayer membrane called plasma membrane or plasma lemma. It is selectively permeable, okay, and maintain the physiological integrity of the cell. Next, cytoplasm, inner to plasma membrane. Cytoplasm is present, which is divided into chromoplasm, outer chromatoplasm, and centroplasm, central region or colorless region called as centroplasm. Generally, the pigments are present in the outer region or peripheral region called as chromoplasm, and central position lack those pigments. They have genetic materials. Thylakoids are present in chromoplasm, okay, and thylakoids have pigments, photosynthetic pigments like chlorophyll A, C, xanthophyll, carotenoid, okay. On its upper surface, phycobilisomes are present. Phycobilisomes consist of 3 pigments like phycocyanin, C-phycocyanin, allophycocyanin, C-phycoerythrin. Okay. These 3 pigments harness the light in sequence of phycoerythrin, then phycocyanin, then allophycocyanin, then chlorophylls. Okay. Next, centroplasm. Centroplasm is colorless, and it is regarded as primitive nucleus because they lack nuclear membrane and nucleolus, so called as incipient nucleus. Okay. Next, cyanobacteria have some cytoplasmic inclusions, okay, like reserve food material called as cyanophycin. Some cyanobacteria have gas vacuoles, gas vacuoles. Next, some carboxysomes, carbohydrate bodies, then phosphate bodies, then phycobilisomes. Okay, these are some cell inclusions. And then heterocyst, these are special cells of cyanobacterial trichome, and these cells are large, pale colored, thick walled cells. Sometimes they appear terminal in position, sometimes they are present in intercalary position, some are present in lateral position. The thick wall is impermeable to oxygen but permeable to nitrogen, because here mucilaginous sheath is absent and photosystem too is absent in those cells. Okay, those cells, heterocystous cell or heterocyst, are specialized cells. They are impermeable to oxygen but permeable to nitrogen, so they help in the process of nitrogen fixation. But photosystem too is absent. So photosynthesis is absent, only cyclic photophosphorylation occur. Because in cyclic photophosphorylation only photosystem 1 is involved. Okay. Heterocyst is dependent for its nourishment on adjacent vegetative cell. It has the enzyme nitrogenase because they help in nitrogen fixation. Heterocyst is specialized to perform nitrogen fixation. Okay. Next, Reproduction, like bacteria, mostly cyanobacteria reproduce asexually, by producing various specialized structures such as actinides, hormogonia, hormocyst, spores, specialized spores. Some cyanobacterial species show genetic recombination, like conjugation, transformation, transduction. Next, there are some vegetative methods. Some cyanobacteria produce spores. Actually, in some books, these methods written as vegetative method, but in some books, there are two separate sections like vegetative method and spore formation. But all of them actually comes under the vegetative method. Okay, so you can write all this as vegetative method. You can also separate these as separate as vegetative and spore formation. Okay, so it is completely up to you, and it is not necessary to write separately all those things separately. You can also write all these things under vegetative method or asexual method. So finally, at first, what is fission method? It is the most common method of vegetative reproduction observed in bacteria as well as in cyanobacteria, that is fission or binary fission. Some shows binary fission where the mother cell or one cyanobacterial cell divide, producing two cyanobacterial daughter cell. In some cyanobacteria, one cell divide and form many cells called as multiple fission. Single cell divide repeatedly to produce many daughter cell. So some cyanobacteria show binary fission, some show multiple fission. Next, fragmentation. Cyanobacterial cell reproduce by fragmentation of their filament or trichome in regular interval into short pieces. Those short pieces actually called as hormogonia, and these hormogonia later on develop as a new cyanobacterial cell, so gliding motility and develop into fully fleshed filament. Next, hormogonia. All filamentous cyanobacteria reproduce by fragmentation of their filament, and the fragments of their trichomes, actually that short fragments are called as hormogonia. Those have the capability to grow as new individual cyanobacterial cell. Okay. Then hormospore or hormocyst. Hormocyst or hormospore is thick-walled multicellular structure formed by few species of cyanobacteria, act as survival mechanism during unfavorable condition. Essentially a resting stage. It can be considered as a resting state that can germinate to produce new filament when conditions improve. It is essentially a specialized hormogonium with thicker sheath, allowing for dispersal and survival in dry or harsh environment. So during unfavorable condition, some cyanobacterial cell produce a thicker wall around it, secrete some substance and make a thicker wall around them, and they can so that they can overcome the unfavorable condition. And when again the favorable condition appear, they again germinate and produce a new trichome. Okay, in this way they reproduce by hormospore or hormocyst formation. Next, what is palmelloid stage? Palmelloid stage refers to a phase or stage where cells aggregate together and become embedded in a common gelatinous matrix. Okay, essentially forming a non-motile colony. And this type of state or this type of structure is typically formed during unfavorable environmental condition. And this is typically analogous, this type of structure is exactly analogous to the palmella stage, which commonly appear in Chlamydomonas. after the unfavorable condition, they again separate from each other and grow as a new individual cyanobacterial cell. Next, some cyanobacteria produce actinides. So most filamentous cyanobacteria develop perinating structure or a dormant structure in adverse condition. These structures are larger than vegetative cells and are equipped with thick wall called actinides. When favorable conditions return, they germinate and produce new filaments. Okay. So these actinides are perinating structure. Okay, and they are generally larger than the normal vegetative cell. But when the favorable condition appear, they germinate and produce new filament. Okay. Then ectospore formation. These are spore develop on the surface of cyanobacteria and they disperse. Okay, they disperse, release as bud, and then survive as a new individual. Okay, on these are survival mechanism typically appear under unfavorable condition, and essentially these are external spore formed by cell division at mother cell's end, unlike endospore which are found inside the cell. Next, nanocytes. These are small cells produced by non-filamentous bacteria in a semi-nutrient environment. They are a type of spore that can germinate and gives rise to new vegetative cell when a condition improve. Okay, so it is also a type of reproduction, vegetative reproduction or asexual reproduction. Some scientists detected or described the formation of planococci in some species like Desmosiphon. Okay, they also grow as a new individual. Okay, so this is all about the reproduction in cyanobacteria. Next is the economic importance. So they are early colonizer on bare land. They are good food source. We can take them as food. For example, spirulina, a filamentous cyanobacterium, is now incorporated in food supplement and called a single cell protein. Some Indian dishes, for instance, puri, idli sandwich, prepared by supplementing S. fusiformis and have been found to be palatable. Next, in some parts of Rajasthan, annabena and spirulina are collected from Sambar lake and used as a fodder and manure. Next, nitrogen fixation is a characteristic feature of cyanobacteria. This function is performed by heterocysts present in them. Ollosira, nostoc, annabena are some such cyanobacteria that are regularly inoculated in rice field. Mainly Ollosira is found in rice field. Okay, this saves the consumption of nitrogen fertilizer. Their nitrogen fixing cyanobacteria are often used in reclamation of osser soil. They produce acidic chemicals for counteracting alkalinity of the soil, and they supply nitrogen compound which are generally deficient in these soils. Species of Annabena and Ollosira do not allow mosquito larvae to grow nearby. Such cyanobacteria can be inoculated in village pond to prevent the growth of mosquito on the stagnant water. Then extracts of Lingbia can be used to manufacture antibiotic-like compounds. Certain cyanobacteria such as Microcystis and Annabena and Aphinozemenon then produce toxin which are harmful to aquatic animal. These toxins prove equally harmful to human drinking and bathing in such water. Okay, so this is one harmful effect of cyanobacteria. Then cyanobacteria generally grow on walls and roofs of building during rainy season and cause discoloration, corrosion, leakage. So these two are harmful effects of cyanobacteria. So this is all about your cyanobacteria. And I hope you really enjoyed this session and will get benefit from this. And please subscribe to my channel to get more important videos like this. So, and please like my videos, share with your other friends. So with this I am ending my lecture video. Thank you.